And welcome everyone who joins us online. Today we are honored to have Marijtje with us here to present and share her expertise and research on pain. I'm going to introduce our speaker a little bit, and before we start with her presentation. So Marijtje is currently employed as an associate professor at the Faculty of Social Science of the Radboud University. That's in Netherlands, yeah, in Nijmegen. She has strong expertise in running event-related potential experiments. She has a, excuse me. Her research interests involve the EEG, ERP recordings, often in combination with neuropsychological tests to study human cognition related to PTSD or post-traumatic stress disorder, the chronic pain, and also anxiety. Marijtje obtained her master in neuro and rehabilitative psychology at the Radboud University. Shall I mention the year, Marijtje? Not needed. After then, Marijtje worked as a research assistant at Westmead Hospital, Sydney, Australia. And after obtaining her PhD degree at the Radboud University Medical Center, Marijtje worked as a postdoc researcher investigating rhythm perception. And then Marijtje in 2002 received a Venny grant. That's a grant from the Netherlands government, right? For a 4-year project entitled It's All in the Rhythm, applying EEG and evoked potential measurements to study cognitive processing of temporal patterns. And then after the postdoc position at the University of Twente, Marijtje became an assistant professor at the Radboud University. And since 2011, Marijtje has been collaborating with several universities in Indonesia with respect to both teaching and research. So in her time here, Marijtje has visited not only UI, but also UNPAD, UGM, Sugia Pranata, and UNPAD, yeah, so, and Undip. So it's been a busy schedule for Marijtje in the past week. And today we are lucky to have Marijtje here with us to share her research, yeah, on pain in particular. I will not be long on this introduction. I will pass on the mic to Marijtje so that she can start her presentation. Would you like to take the stage, Marijtje?
That's so great. Okay. Thank you very much for a very nice introduction. You didn't introduce yourself, so I can return the favor bit. Kemala. Yes. Currently doing a PhD at the Radboud University.
Yeah.
As funded by LPDP. And the rest of your team.
Okay.
I know it is.
So let's go back a bit. My name is Kemala. I'm a staff here at the Faculty of Psychology. I'm a member of the Lifespan Development Research Group here at the faculty. Oh, we have our member here, Ibu Ika, welcome. And I'm currently running my PhD studies, both in the Netherlands and in Indonesia. Yeah, so that's a bit about me. I will be the moderator of this talk, and so we'll have later questions session and some demonstration to show you how pain is processed. Or sort of, we'll see how we can elicit pain. Yeah. So, Marijtje.
Okay, thank you and think judging by the audience, you might actually already, already told them maybe that they can get some experience in being in a pain experiment as well. So we're all introduced. We could actually even make a round, but I think we can believe it. So what I would like to do in this research seminar is to first, I mean, it's unavoidable. I will have to go into the definition of pain. And that is not as straightforward as you might think. The people who are going to the lecture tomorrow as well, this will be double. But any lecture, we have a whole course on pain. And every lecture in that course, I begin with the definition. Can we administer pain in the lab to study the effects? And how can we do that? And is it really pain? And is it ethical? How can we actually measure pain? So the pain response, the pain perception. Or can we maybe also study pain without using painful stimulation in the lab? And how can we study how our brain and mind may change the pain experience? And this is all very much related to the definition of pain. One of the introductions, we do have a full course on pain perception and treatment in February and March. That's also open to a selective group of students from our partner universities in Indonesia. It's a joint course with Pajajaran. And I think there's still some openings if you like to have more than 1 or 2 lectures. Now, I think a lot of people, when I ask, what is pain? I mean, I can ask it here. Anybody care to? They might actually come up with a definition very similar to this one. And this is actually taken from a Dutch dictionary on pain for general practitioners. And it states that pain is the unpleasant sensory and emotional experience. So it's not just sensory, but we also have label an emotional experience to it, due to the strong stimulation of the end of sensory nerves or pain receptors as an elementary warning warning of imminent danger. And that is what we know as the tissue cable model. It states that in order to feel pain, there must be some kind of injury or stimulation which will result in the pain. It's known as the tissue damage cable model. That has also been postulated already in times of Descartes, where you're being threatened by a noxious stimulus or damaging stimulus. In order to protect yourself, you have to withdraw from that. That's very basic survival behavior as well. However, this definition is not correct, disclaimer, because there's not really a one-to-one relation between painful stimulation or threatening stimulation, damaging stimulation, and the experience of pain. We can, for example, have tissue damage without experiencing pain. So that it depends really on the situation. So we might be threatened and in danger, even injured. And one of the most fascinating examples is stress analgesia. And this can really take extreme forms. This is an example of Aron Ralston. And Aron Ralston is an American mountaineer. And there's even a movie made out of one of his experiences. He had been mountaineering on his own, made a fall, and actually his arm got trapped under a rock. And he was really stuck. Probably his arm was broken as well. And after, I think, an extended period of time, he started to realize, he was all by his own, that he would not be found and he would just die there, you know, if you wouldn't free him from under that rock. And the only way to free himself was actually to cut off his arm. And he had a pocket knife, most climbers have, and he decided at that moment to cut off his arm. And he managed to do so. And he was often also, I mean, that must be like extremely painful, but he was so observed in the task at hand and even fascinated the way he described it, just, you know, just really focused on the task, hacking through the tissues. He said only when he had to cut through the large nerve fiber, that he felt an overwhelming sensation of heat, like hot lava. But apart from that, he just was very rationally doing what was needed to survive, got in the vicinity of a road afterwards with just a bottle of water and was rescued and survived the incident, giving inspirational talks and still mountaineering with a prosthesis. But showing that under extreme conditions, we can completely block our sense of pain. Yeah, so there's not a one-to-one relation between the type of stimulation or injury and our experience. We also have the opposite, and I like these kind of experiments. They have also done in a scanner, but that is the phenomenon of pain empathy. And it's actually when we see people that are in pain or make a fall, our pain empathy actually activates similar brain areas that are also activated when we experience pain ourselves. It's even if somebody close to you is, for example, needs to have stitches, some people physically feel this similar pain. Yeah. And they even did it in a scanner. I don't know if the movie will be working. I will try that. Yay!
Recently, a number of research have shown that when we perceive someone in pain, someone being hurt, we activate in our brain the same network, almost the same circuits that will be activated when we are ourselves in pain. So there is a sort of overlap between perceiving pain in others and experiencing pain in oneself. That's what we call empathy for pain. This mechanism of perceiving others in pain using the same neural machinery that we use for experiencing pain has probably evolved to detect and move away from sources of danger. Now there are situations in which one has to inflict pain for the sake of the others. Think of situations that nurses, dentists, physicians, surgeons have to cope in their everyday life. They have to inflict painful procedure for the sake of their patients. So in this study, what we did, we had two groups of participants. One was a group of control, non-expert, and the other group was a group of physicians who practiced acupuncture for a couple of years. In the scanner, they were watching body parts being pricked by needles. In the first group, the group of the control participants, we found activation of the pain network, the pain empathy. But that was not the case for the group of doctors. That network was not activated. Instead, regions of the brain that are involved in emotional regulation and cognitive control were activated to inhibit their sensitivity to the pain of others. In addition, doctors had a tendency to report less feeling of unpleasantness when they were watching the needles than the control group. So altogether, that study shows that doctors need and have to regulate their sensitivity to pain. They have to regulate their empathy for pain in order to be of assistance for their patient.
I like that because, as you might have picked up from my CV, I did my PhD at the Department of Anesthesiology in the hospital. And we have been discussing ever since if this phenomenon that doctors don't really have pain empathy, or not in a way that non-doctors have, if this is already a trait, so you're more likely to become a doctor if you are not really having that much pain empathy, or if it is learned. So if we would study this in every year of the education, what we would find. But it mirrors quite nicely the previous finding. So again, there's not a standard response either to perceiving pain in others, so pain empathy, or in actually having injury leading or not leading to a certain pain experience. So it depends very much on the situation, the person, and also previous experiences. Now, what I like about the pain empathy is that this is a research seminar that we can do a little experiment with it. But first I come back, so this initial definition of pain, the tissue cable model, actually refers to what we know as nociception. And nociception is the perceptual phenomenon of your ability to perceive damaging stimuli. And it comes from the Latin word of nocere, injury or hurt. So it's our ability to detect tissue damage or the threat of tissue damage, potential tissue damage. Now, what is pain then? What is the current definition? And if you really truly understand the definition and the difference between pain and nociception, I tell our students, then you have already mastered half of our course in pain. Until 2020, the formal definition was that pain is an unpleasant sensory and emotional experience that is associated with actual or potential tissue damage or described in terms of such damage. So you can even experience pain without an actual stimulus being there. And actually the majority of our pain has nothing to do with a noxious stimulus, with a painful stimulus. Just think about chronic pain or headaches or migraine. Yeah? However, it has the word described in it. And that actually excludes a lot of situations where people are not acknowledged to be able to experience pain. For example, if they don't have verbal capacities, or if they are, for example, this is a patient in coma in the hospital. Infants, so I think until the 90s, it was presumed that infants did not feel pain because their pain system had not yet developed, which is, I mean, horrific to think about, that even during surgeries in infants, they would just give sedation and muscle relaxant, but not really analgesic medication. So they updated that only in 2020, and nowadays the formal definition states that it's an unpleasant sensory and emotional experience associated with or resembling that associated with. So you can also determine that someone or a sentient being is experiencing pain by observing actual or potential tissue damage. So it also states the end that if you have tissue damage, but you're not emotionally, it's not an emotional experience, it's therefore not pain, if it's not aversive. Yeah? So it's really coupled to the emotional. Now, just to do a little check, this is the same situation, or this is the same patient in two different moments in time on intensive care. When would you increase pain medication? Up or down panel? Everybody, you can point up or down. Which picture is the patient in pain? Up, yeah. So in general, and this is, I mean, this got only acknowledged in 2020, whereas most of the time everybody has no difficulty in determining what a person in pain looks like. I still find that actually quite shocking, because it also says something about standard protocols in nursing and pain management. So yeah, here's the typical behavior, and there's now a lot of observation skills as well that are being used in hospitals with raised eyebrows, higher cheekbones, etc. Now, if you look at both definitions, so the definition of nociception and pain, we can see that they actually mirror each other in a way. Nociception can be determined also from a third-person perspective. It's clearly stimulus-related and has also strong links with sensory discrimination. However, pain is first-person perspective. It's a perceptual, emotional phenomenon, and it is closely related to the concept of suffering. Now, nociception is something that we can mimic in the lab. We can even do it here in the demo. And pain can result from nociception, but it doesn't necessarily have to arise from nociception. And also pain can arise without nociception. Well, there's a little bit the summary of that one, of the definitions. In order to have a pain experience based on nociception and get to suffering, also the social context plays a large role. And we've seen that in the example of stress analgesia. Depending on the context, you're going to act or suppress your pain, or maybe even increase your pain sensitivity. So nociception we can see as the tissue damage cable model, but that's not just a passive one-way direction. It's not just that pain information is being sent to the brain because the brain responds to it, and there's also descending projections that can modulate this pain experience or the incoming pain information. So again, a nociception is a neural process of encoding this information, and the consequences may be autonomic. So for example, an increase in heart rate, elevated blood pressure, withdrawal reflex, or other nocifensive behaviors, protective behaviors for painful stimuli, but not always pain. And nociceptive pain, on the other hand, we reserve for situations where pain is clearly triggered by a noxious stimulus. And you can maybe easily see how we can study that in the lab. So there are actually a lot of interesting ways that I get paid to do to my students. And that is to inflict controlled and harmless pain and then ask, did that hurt? I really enjoy my work. So we have, you can actually do that with either heat and cold stimuli. We have a demo of that one. We have the von Frey filaments, which are pretty much very thin filaments that get thicker with every step, and you measure when, so it becomes more and more inflexible when you put pressure on the skin. And the thickness of the filament determines the pain sensitivity. The algometer is a pressure, so measures the sensitivity for pressure pain. And I always like, this is a very simple demonstration, but if you just get your muscle of your thumb between your two fingers, the belly of the muscle, then you can also feel that your touch system is different from your pain system. If you start to squeeze it, you'll just feel the squeeze. And if you continue, there's a certain moment where the sensation suddenly changes from just pressure to pain. It becomes actually very unpleasant. And that's a very clear and specific moment in time that you can distinguish, and that is your pain threshold. Do you have difficulties finding it? Because I'm happy to squeeze you. Huh? Yeah. Just do it very slowly, not just repeatedly, but slowly, and just keep increasing the pressure. Okay. I find it quite easy to find my pain threshold that way. This is touch. Now tell me when the pain starts. Yeah, okay. Stress analgesia. Congenital insensitivity to pain might be. We'll check your pain empathy in a minute. Normally, it is quite easy that way to find the pain threshold. I'm sorry about that. And then the electric shocks. And we do that a lot because it's also very easy with electric shocks to elicit an event-related potential in the EEG. Now, what's the most reliable way to measure pain? The most reliable most reliable way to measure pain is actually to ask the person how much pain it does, because by definition, pain is the perceptual and emotional experience. So the benchmark is what a person reports, so self-report. Now, the funny thing is that when I did my PhD at anesthesiology, the whole aim of my PhD was actually to find an objective measure of pain. And by definition, that's not possible because pain is not objective, not third person. One of the most sensitive ways we do it is just with a visual analog scale or a numeric rating scale. And if you, for example, use one of these tests, so heat, cold, pressure, electric shocks, you can start at a very low level and first ask, do you feel something? And the moment people start to feel something, they can rate it as 1. That's your sensation threshold. Yeah? It just says something about the ability of your somatosensory system. Does your somatosensory system has to be intact or fully functional to feel pain? No. And we see this in certain neuralgic syndromes. But for example, you can have this experience yourself also. Everybody knows the experience that if you have been sitting down too long and your blood flow is restricted to your legs, for example, you stand up, your leg is numb. And if you, your sense of touch is greatly decreased, but if you pinch it, actually your sense of pain is increased. So you're more sensitive to pain and less sensitive to touch. And it has to do that different fibers are involved. For touch, the information is mostly projected via thicker myelinated A-beta fibers. But with pain, it's more the A-delta, A-delta, and C fibers, unmyelinated thin C fibers that become unmasked. The next step, if you increase the intensity of the stimulus, then at a certain point people say, normal people, they will say, okay, here, this is where it starts to hurt. Yeah, if you increase the pressure. And that is the first moment of pain, which is your pain threshold. And there we know, this is where the pain system starts to become activated. And information is now also able to activate A-delta C fibers. And then comes the fun part, because it also depends on report. You ask, can I still increase the level until the maximum you are willing to have in an experiment, your pain tolerance? Yeah, and that's where you go. And of course, people have to trust you, but you try to find the pain tolerance level. And as long as people know this is safe, they're so curious in finding their pain tolerance that they actually come with quite high levels. Okay. If you look at the brain and if you look at nociceptive pain, what we see in acute pain is that ascending information travels via the thalamus, and we see increased activation in the primary motor somatosensory cortex at first, but also in the insula. That's important for pain perception, nociceptive pain perception, but also related areas. So the amygdala gets activated, the cingulate cortex and the prefrontal cortex as a response to also control your response to the pain or act upon it. also known as the pain matrix, although there's a lot of criticism on it, because the same network is activated in a complete wide variety of salient stimuli. So nowadays, they rather refer to it to the saliency matrix. And it's not just reserved for pain. Now, the interesting thing is that if we look at the pain system, and this is a simplified version of ascending information, so the noxious information that's being projected to the brain and the reaction of the brain, there's also activations that contain descending information. And that works a little bit like this. If the pain matrix is being activated, in general, your whole brain will start to create a modulating response via a pathway that also goes through the thalamus, has a strong connectivity with the periaqueductal gray, which is a key area within our endogenous opioid system, involvement of the rostral ventral medulla with on and off cells for activating, deactivating the incoming information. So at different levels of the system, incoming pain information can be modulated, can be gated in, or can be suppressed. Now, we did one study, I'm not sure if there's people with a huge fear for dentists, but this was actually one experiment that we can also try here. And that's an experiment with pain empathy. There's a little bit of background. We have 2 movies, and we actually had 2 different groups. So we collected some data from a complete group. One that was watching the control film, which is just an instruction how to brush your teeth. And one is a root canal treatment of a girl that posted it actually herself on YouTube, that didn't want to have anesthesia because she was more scared of the anesthesia. There's a lot of screaming in the second movie. Of course, it's a little bit more distracting. But that's just one part. The other one is a very easy way to measure your pain tolerance. Now, if we combine the information from the first study with the pain empathy, that is, if you see people in pain, your brain is already activated as if you are experiencing pain yourself. So your pain matrix is activated. If your pain matrix is activated, we see a very interesting phenomenon in general, is the phenomenon that pain inhibits pain. Yeah? If you hurt yourself, you start to activate your pain suppressing mechanism, making you less sensitive after that for a new noxious stimulus. Now, how we normally measure that is we determine somebody's pain threshold, either with pressure pain or electric pain. Then we have a longer exposure to a tetanic painful stimulus, which is exposure to ice water. And then after, this is actually very painful. This is what we can try out here. So do I need to put some water in there, Jude, or not? Oh, there is. Oh, yeah, it should be half water. Half water, half ice. There's half water. Yeah. And, maybe just, yeah. Okay. They'll get the ice water. This is a very easy test that's also being done in the hospital. If you fill a bucket half with ice, half with water, you have a very stable temperature of around 4 degrees. And if you put your limb in it, it's harmless, but after a while, it's really, really, really painful. And it's very easy to measure. You just take your stopwatch. You ask somebody to stick their hand in, keep their finger spread like hanging it loose, and see how many seconds they can actually stick their hand in. You have to instruct them, because if you know this test and if you just stick your hand in, And you know at a certain point, in the beginning, the pain will increase very quickly and then it sort of levels off. And then actually it becomes doable again. So if people train themselves, they just keep their hand in and they're okay. So you really have to ask, now take your hand out if you reach that maximum. Now, if you do, so we can try. Who would like to see what? Yay! We have a volunteer. Do we have a stopwatch as well? Oh, maybe you can do it here that people can see it as well. Oh, yeah. We can do the stopwatch. Do you want to watch the film at the same time? Because then you should be able to keep your hand in there longer. Are you scared for the dentist? No. We do it with it was no, it was here. Sorry. We do it with the dentist movie. And we started and after you, because you have to watch it first to activate your pain center. Then after 10 seconds, we ask you to stick your hand in the ice water. Yeah? Yes, okay. And you have a stopwatch? Yeah. Yeah, that's not nice. So nice and deep in there after when I say yes. Yeah? You ready?
Ik moet stoppen mogen gerust zeggen. Dus achterop dat ik het kan zien. Moet het verdoven?
When the pain is maximal. That's pretty long, 29. Yeah, but it starts to hurt. Yeah. But at the same time, it's quite a safe test. Now, I'm pretty, because you are really watching the film, of course, there's a lot of confounding variables here, yeah? Because in the other room where we had this movie,
at like somewhat of like a 90-degree angle, and you're going to place it, therefore the bristles are touching your gums and your teeth, and then you want to go around in a circular motion. What you do not want to do is go back and forth or go up like this, because that will actually cause...
It's just very boring, so they also differ too much in excitement, yeah? So arousal. But is your hand okay? It is supposed to be completely safe, and it looks like, yeah, ice water is around everywhere. But you do get a lot of very nice results by measuring pain tolerance with a very simple setup in different situations. We also did some very nice small student experiments with it. Also, for example, in football players, so the effect of winning or losing, team spirit, etc. We had a lot of fun with it. Anybody else care to see the pain tolerance? Anybody that can beat the 29 seconds?
Ya, terima kasih Bu. Ini.
Yeah, sure. Who's having the stopwatch? No, I think we've seen the movie. One, two. Yeah, yeah. We just now when it reaches the maximum, it ramps really quickly, right? Yeah. And I can start my heart rate faster. I think that's how it's supposed to be. Yeah.
You know, in the Netherlands, we go by bike, backwards, in the morning, in the winter.
Your forehead, yeah.
Like that. But I can feel my heart beating faster, and my breath is getting. Is this also related to gender differences?
I always get this question. There is a difference. We did some gender experiments as well. In general, men have a higher pain tolerance, so they can have more pain and also higher pain thresholds. But women show a higher capacity of suppressing the pain. So in second pain, they last longer. We found in general just a little bit higher pain modulation. Not yet.
All about mine.
Eating chilies for lunch, that might have.
I think you get used to the.
But when you get used, you're actually past that maximum. Yeah. Then you're past. Yeah. And then we also ask a rating. So how painful was that in general on a scale from 1 to 10? Okay, I'm very atypical in my participants, or you're just, yeah.
This is because I don't wear gloves in the winter.
Yeah. What I like about this little, so you already see there's a lot of personal variation. But what I like about it, it's used in clinical settings, and it actually does have quite good predictive value. And I do it in pain centers. And there's one clinical study that have found that if people are not very capable, so normally you use the ice water to increase your pain threshold before and after ice water. So after this exposure, if I would now check your sensitivity for pain, you would be less sensitive, showing that you are capable of activating your endogenous opioid system, releasing your own painkillers. Yeah. Now, your capacity of doing so is predictive if risk for developing chronic pain or not. So people who are not really capable of increasing their pain threshold have a much higher risk to develop chronic pain after surgery. So it has clinical relevance, and that's what I like about it. I was asked to include a demo in the seminar. And what always amazed, there's actually one funny story about the root canal. I did also this seminar for our department in the hospital, and there was one surgeon, a surgeon that I wasn't particularly fond of, so a real typical surgeon. And apparently it's really profession specific, because he could not watch this film. He almost crawled under his table, and he said after 3 seconds, yeah, we get the idea. Just continue with the talk. Just continue with the talk. It's just, whereas, you know, you could show him surgical pictures and movies to the max, but this dental movie, he just couldn't deal with. Sorry, we have seen that as well. Okay, so to summarize, we've seen that actually the self-report measure is the benchmark. It's the most reliable. But if we want to say something about the direct response of the brain, there's also other approaches. So if you look at the information processing theory by Miller, then if we have some kind of stimulus input and any kind of stimulus, there's information processing going on, and based on information, we might generate a certain response. Yeah, for example, rate the intensity of the noxious stimulus. In your cases, I usually go to 8.5 on ice water. Okay, but still it classifies as pain. the problem is between the moment of presenting a stimulus, processing that stimulus and generating a response, there is a lot of noise happening, interference, that might also additionally affect the kind of response we're generated. We might be distracted, we might experience social pressure, etc. So then I come back to the reliability question that I was initially asked. So although this is the best measure for the pain experience, still, to see how the noxious stimulus is being processed, we would like to look directly by means of neuroimaging to the response of the brain. And in this kind of setups, you can really, by comparing both the direct response of the brain and the consecutive pain, self-reported pain rating, can actually deliver some very interesting results to get a better understanding of pain modulation by the brain. So the way how the brain is capable of top-down modulating our pain experience. Now, if you look at the different pain imaging methods, so in the first movie, you already heard that pain empathy has been studied with MRI, showing that this overlapping brain areas are activated when we see people in pain or if we experience pain ourselves. But there are other ways, and this is just a standard picture of neuroimaging. And all these different neuroimaging methods differ pretty much in the way they have a temporal resolution, so how accurate they are in saying where something in the brain is happening, or in their or the temporal resolution, spatial resolution. Temporal is how high the time resolution is. If you look at the EEG, it has an incredibly high temporal resolution, though a fairly low spatial resolution. But when it comes to information processing especially, and that's the game I'm in, then having a very high temporal resolution is interesting because you want to monitor on the millisecond what's actually happening, because we know that there's actually a lot of consecutive processes between the presentation of a stimulus, in this case a painful stimulus, and the generation of a response, for example, a rating. The stimulus is first being picked up by the primary cortex, then it's going in working memory, it's probably being compared with some long-term memory. Areas are involved in analyzing the stimulus and labeling it, etc. Now, here we see the EEG. This is actually a setup with a mobile lab during a festival where we did also a pain experiment as a demonstration. So it's also quite an accessible and easy method compared to other neuroimaging methods. You can set it up in a short time, and and it gives you a lot of data in a short period. That's what I like about it as well. The thing is, if you do a full recording and you present people with pain, there's not so much you can tell about the information processing. Yeah? I mean, basically what you end up with is a lot of wriggly lines on the screen. Now, one way to extract the information, the way the painful information is being processed in the brain, is by extracting the event-related potential from the ongoing EEG. And that works as follows. If you present somebody several times, for example, with an electric stimulus, painful stimulus, then every time you present it, you will take the surrounding EEG, and if you average a couple of trials, let's say 10, 15 trials, all the activity in the EEG that is not really related in the processing of the stimulus will average out, will on average be 0 because it's random. But the information or the activity that is time locked, so always at the same moment in time after the presentation of the brief painful stimulus, will remain visible. And this is what we call the event-related potential. And one of the advantages of working with painful stimuli is that they actually give a fairly high response in the EEG, and that you only need a couple of repetitions, 5 to 10, to already be able to extract a stable event-related potential from the EEG. Now, if we zoom in on this event-related potential, or ERP, we know that the early components are mostly determined by the physical stimulus characteristics. So, for example, if you present sounds, these first components will be determined by how loud the sound or how high or low it is. And the later components, they're also called the endogenous components. They are more related with the later information processing capacities in the brain, and depend very much on the meaning that's allocated to the stimulus, or attention. So they are very much endogenous. They're called the endogenous component. The first one, the exogenous components. So these reflect basically the ascending incoming information, and these are more associated with the final perception, appreciation, and experience of the stimulus, the conscious experience. So we now have actually very interesting measures to start to measure the pain during an experiment. We can ask the person how much was the pain, and we can also look at the event-related potential associated with that painful stimulus. Now again, we had a look at the endogenous analgesia. So we did the ice water test in an experiment. And here we looked at the conditioned pain modulation, the test that I explained that they also use in hospitals to see somebody's vulnerability for developing chronic pain, or the ability of somebody to suppress pain based on pain. So what we did was first determine the pain threshold and the pain tolerance with an electric increasing pulse. So you first get a very low shock, and then the shock will increase. After that, somebody had to stick the hand in the ice water as long as possible. And then immediately afterwards, we would measure again the pain threshold. Now, the percentage increase in threshold reflects your capacity of activating your endogenous analgesic system. So your endogenous. What we see if we use a standard painful stimulus, and for the ERPs I want it because I want the exact same level before and after the ice water. Because then, if it would only be determined by the stimulus characteristics, you would see the exact same evoked potential. So this experiment, we actually found a level that a person would say, okay, this is what I can have. We actually had two levels. We say pick a high and a low level. So they wouldn't actually know after the exposure what kind of stimuli they will be getting. And we had them rate the painfulness of the stimulus before and after ice water. And we see that as the stimuli, so painful stimuli, the shocks were rated lower after ice exposure than before. And we did it even counterback. So we had ice water and room temperature water. And after a baseline, people would first be exposed to either room temperature water for 2 minutes or the ice water, and then test again. Now, as said, and here we see the whole mechanism. The conditioned pain modulation depends on activation of the endogenous opioid system. How do we know that? We know this because we can easily block this system. If we present people with naloxone, naloxone is also used if somebody takes an overdose, for example, in heroin or other opiates. It blocks the opioid receptors, making your body insensitive for any kind of opioids. Now, they have seen in similar experiments that if they give people naloxone, they will not show this conditioned pain modulation effect. Yeah? So their sensitivity remains exactly the same after ice water exposure than before. The key structures that are involved that we know from neuroimaging are the periaqueductal gray that we know is the key area in the endogenous opioid system, and the RVM, so the rostroventromedial medulla. As said, it has clinical relevance. And we also know that this system is not fixed. This system shows also neuroplasticity. So depending on certain experiences, your activity in your endogenous opioid system might become higher or lower. And actually, and this is what I really like, quite recently they have developed a novel treatment, for example, for a chronic pain syndrome, fibromyalgia, where they gave participants a very low dose of naloxone for a period of time, with the idea that maybe the system is being exhausted. Yeah, if you have to use your opioid system constantly, it might become less and less sensitive, almost like a rubber band that is losing its by shutting down the system for a period or partly shutting it down, the system might start to sensitize. Yeah, so the neurons that are involved in this circuitry might actually start to sprout more branches and synapses and become more sensitive because they're seeking input. And what you see is after a while, actually, those patients start to experience less pain, although they're taking a drug that shuts down the system and maybe increase activity. Back to the experiment. So we measured the evoked potentials in a conditioned pain modulation experiment, actually during a festival. Again, we had the two conditions, room temperature and ice water. First, the baseline ERP on a step and a pain rating. As said, there was actually two different levels. And then we measured the ERP after temperature exposure. You see a significant, I think this is a little bit, we had it, I think, Friday. Work, yeah, it's going on and off. There might be a little bit of a repeated testing effect, but this is the nice thing about counterbalancing, that we took care of that. A little bit decreased, but after the ice, we see that not only the rating is decreased, but also a later component of the potential that we associate with also this decreased experience. So we can demonstrate this effect, this conditioned pain modulation, not only in the appreciation of the painful electric shock, but also in the direct response of in the EEG. Now, to make it even more interesting, apart from these, so we showed this is an effect that we can measure both with a self-report measure and with a neuroimaging method, but we also took some questionnaires in the participants, asking them about their pain sensitivity. And there's many different questionnaires, but we looked at somatosensory amplification scale. And that is a questionnaire that basically asks how sensitive are you for any kind of somatosensory experience. And this questionnaire consists of statements like, if somebody is smoking in my environment, I easily start to cough. Changes in temperature, etc. Now, what we see now is that there was like no. You did find a very nice correlation between the tomatoes. Shall I use that one? Friday, so that you can be ready to go into this. Yeah, there might be a risk because I remember on Friday we changed and related the batteries as well. So I see a minor flaw in this approach. So we did observe a correlation between the somatosensory amplification and somebody's ratings on the baseline already, so on the pain sensitivity. But what's maybe even more interesting is that also this capacity to suppress the pain, so the activity in the endogenous opioid system, also dependent or also showed a correlation with the tendency of somatosensory amplification. So the more sensitive, the less people were capable to suppress the initial pain with ice water. So these pain attitudes are not just a phenomenon if you exaggerate or not, or, if you're, as we say, like a sissy, like very sensitive for pain, but it's probably also dependent on your physical capacity to modulate pain and to suppress pain. Now, a related phenomenon, we still have some time, right? And what we also did was we looked at the placebo effect. Probably everybody's familiar with placebo effects. Rings a bell. Maybe a little bit less well known is the nocebo effect, which is the opposite. So if I have, for example, a pill and I say, okay, this is a very good painkiller, whereas it does nothing, it will actually, in a lot of people, give a decrease in their pain experience due to the suggestion. Even nowadays, they've discovered if you do that open label, if people know there's no active substance in the pill, they can still experience a pain reduction based on an open label placebo. The nocebo effect does the opposite. If I give you a pill, and even if it's not having any active substances, you might report an increase in side effects. Like, okay, I always get nausea straight after I take it, and you can even give a suggestion. Yeah, and nocebo effects are as relevant, but they work with via a different neurotransmitter system and mechanism. What we know about the placebo effect is that, as with this conditioned pain modulation, it is capable of activating these same pain inhibitory mechanisms. And also, like the conditioned pain modulation, the placebo effect can be reversed or stopped by administering naloxone. So we know that the placebo effect also depends on the endogenous opioid system. There's also a very important role of the cingulate cortex and the dorsolateral prefrontal cortex and the anterior insular cortex that we know in general play a very important role in controlling behavior, regulating behavior, also controlling emotions. And there's a lot of individual and clinical variances that are linked to these differences. Now, how do we test that in the lab? That's actually quite. If we use just a stimulus target response task, yeah, then I can tell people, okay, I give you 2 different kinds of electric shocks. Half of them will be on your personal level 6, and the other half will be on your personal level 8. I set the levels personally. Every time you see a green square, you get a very painful stimulus. And if you see a blue circle, you get a less painful stimulus. Yeah, stimulus response. Very easy, very quick experiments. And as a psychologist, of course, in 15% of the occasions, we change the warning signal. So we say you're actually getting a high intensity shock, but giving them a low intensity shock. And they expect a low one, and we give a high one. This, of course, will induce a placebo-like suggestion. And this one, expecting a high shock but getting a low shock, induces a nocebo suggestion. So the placebo-nocebo-like effects. Now, what happens? As with the previous experiment, we see so this is more like a replication because this has done many times. We see that the appreciation of the shock follows the suggestion. So the painful shock is experienced less painful if they expected a low shock, and the low shock is experienced more painful if they expected a high shock. We can also see this back in the event-related potential. It's a very subtle effect, but it's significant. But there's a difference. What we see with the placebo effect is that, oops, help, that the highest effect is actually frontal electrodes. And we see that in a P3 effect, whereas the response to, so keep in mind that the shock is exactly the same, right? In both cases, it's the shock. But for the correct information, they expect a high shock and it occurs. And for the placebo suggestion, they expected a low shock, but they got the same high shock. But the response, especially over the frontal cortex, is now decreased. We see the opposite with the nocebo, but more posterior. If they get a low shock, but they expect a high shock, they have an increase in the P3, and they show also mostly over the posterior. And this can be linked to the effect, but it's a little bit more complicated because when we started to do this experiment, we knew that the nocebo effect and the placebo effect actually rely on different neurotransmitter systems from the literature. Placebo suggestions are mostly explained by activations in the endogenous opioid system. So the opioid receptors play an important role. But if we look at the nocebo suggestions, we see that another system is more important via anxiety. We know that the system, cytokines, they play a very important role in explaining this nocebo effect. And then one of my, dus cholecystokinin. We know that there's one very important center in the brain pain mechanisms that's low in the brain, ventral medulla, that I showed a couple of slides before, that contains of cells, and these cells are inhibited by opioid, probably suppressing incoming noxious information, but they can be activated at the same time by cholecystokinin, increasing pain sensitivity involved in the nocebo effect. Because these two systems actually have an effect, an opposite effect on the same kind of cells, one of my colleagues said, well, in most standard placebo-nocebo experiments, the effects are always mixed, but maybe it's better to investigate them in isolation. So only induce a placebo suggestion in one block and a nocebo suggestion in the other block, so you get no interference by on and off activating these two systems. Does that make sense? I'm here at risk of losing some of the... Exactly what we did, but participants did not realize that. So, again, counterbalanced. In total, participants would get 100 stimuli, 100 trials. 50 with a high shock, 50 with a low shock. In the first block, however, we would give 25 cues. There was no 200 shocks, 50 cues for a high shock and 50 for a low shock. But with the low shocks, 15 were followed by a high shock and the others by a low shock. It's the placebo suggestion block. After they would get the placebo block that contained a little bit of shocks. So the blocks were determined on the cues. Yeah, there were always 50 cues signaling for a high stimulus and a low stimulus. Only in the block there appeared incorrect low cues, and only in the no block there appeared incorrect or unreliable high shock cues. Now we actually get 3 different kinds of stimuli. Shock are correctly cued in the placebo block, but they are very reliably. And we get high shocks that were preceded by a cue that a low shock should be expected. And in the block, they are seen as filler trials, the high shocks. And they were less reliable predicted. So in the placebo block, we have 50 low cues, 35 correct, 15 incorrect, and 50 high cues, all 50 correctly cued. So a little bit more high cues. Now, to summarize, what we see is that the placebo effect and the nocebo effect, but also if we compare to the more general, constantly activating either the endogenous opioid system or the CCK system, effect or increasing pain sensitivity and also response. So within block effects, which are effects of the suggestion, but we also which are the general placebo nocebo state by constant activating of either the endogenous opioid system or the CCK system. And the better. Okay. Now we go one step further. Experiment, we also looked at pain attitudes. So again, we took a lot of questionnaires. Yeah. So we asked people about their hypochondric tendencies, about experience, health, pain catastrophizing, which is a tendency to amplify the pain itself and to allocate more negative associations with it. Sensory education. And these pain attitudes actually also have an effect on these descending pain pathways. And what we see is that the netto effect going from the pain experiment or suffering is also being influenced by our expectations, past pain experiences, also biological, physiological uptake, state at the moment. Now, these were the kind of questionnaires. So again, the somatosensory amplification. Somebody have two. Noises disturb me. Silence is I'm very. They're constantly being exchanged with each other. Swap with the word last for 5 minutes.
How about we have a quick break? I see that everyone is in painful condition. So maybe stretch our body a bit, our legs, or if you would like some tea for the people who join us offline, we have some tea or coffee, I don't know. But yeah, please go ahead. For people who are joining us online, we're gonna have a short break while we're trying to figure out whether the technical issue has been solved or not. And then we'll be back again to continue the lecture. How long would you like the break to be? Do we still have that? Okay, so about 10 minutes? Yes? Okay, please feel free to have some tea. People who are joining online, let's have a 10 minutes break and then we'll come back. Thank you.
Ja, ja. Goed om goed goed te beluisteren. Ik heb eigenlijk een project over fijn. Heel dus dit is gewoon fijn is ook zo. Laten we niet met de versie van tegen op de kant. Oké, dus wat ken je steken dan? Is het wel heel goed aan op mijn werk. Zo heet dat. Maar dit willen zijn er ook mensen die bezig zijn? Wie zit? Ik zit nu in pijn. Heel specifiek dat. I don't like the fijn. Zo. En ik kan niet. Oké. Dan ga je best wel. Dan heb je best wel. We hebben het van zo'n fijn tegen. Dus maar mijn gewoon fijn weer het andere goed. Ja. Hij wil ook ja, dat is niet groot is, maar gewoon ja, je hebt natuurlijk wel eigen film, maar dus je wil eigen films, die krijg je ook van. En dan samen met kunstenaars dat je echt dat wel geld voor aan hetzelfde.
Well, we're having a break. Maybe if you have some questions that you would like to ask to Marijtje later on, you can go to the menti.com and then enter the code. It can be any questions you're curious about. Maybe you find some pain experiences that you've experienced in the past that you find it a bit unusual, that you would like to find some explanation about your experience, then maybe we can talk about that. Or it can be research-based question that you would like to know more about the methods used to elicit and modulate and measure pain. That can also be your question. So any question is valid and we can try to address it. People who are online, please send in your questions through Mentimeter. So the link is already I think it's available on the chat window, and you can just click the link and enter the code and type in your questions there.
Ja. Middag.
So apparently there are some problems with logging into Mentimeter. Apparently you have to log in with your account, and that's a bit challenging for some people. So we'll just skip the Mentimeter and just throw in your question in the chat window. It can be in Bahasa, it can be in English, because Marijtje is very fluent in Bahasa. Yeah, so she can practice her Bahasa by answering your questions. Yeah, just feel free to use Bahasa or English, whichever is more convenient for you. Or Dutch. Perhaps only Dimitri can send in questions in Dutch, but who knows? For people who are online, we had problems with Mentimeter. Please just send in your questions via the chat window. Or maybe we have time a bit later on, and you can speak and ask your questions directly to Marijtje. Yeah.
Are we now in the break still or in the question period? It's not already started.
Still in the break. You already have questions.
No.
Seems that no one is getting tea here. So everyone is too relaxed in their seat. Then we'll just digesting their lunch, yeah. So we'll just get started with Marijtje's talk. Yeah, continue. Yeah, Marijtje, you're done with the tea? Yep, okay. Go ahead.
Good. I'm going to I was almost finishing this placebo-nocebo experiment. And that is, we linked it also to the questionnaires. What we found. So this can be a little bit challenging to follow. But remember that we had an induced placebo and nocebo effect, and more a state effect. So this was the effects within a block. Yeah, so the direct suggestion of a placebo or nocebo suggestion. And this was actually if we compared the high stimulus from the nocebo block with the high stimulus from the placebo block as the filler trial, and the other way around. Now, first of all, the effect of the state. So continuously in a block, there was an effect, but very small, and we did not find any correlations with the questionnaires. But if we look just at the pain scores, so for the high shock, so these were all the scores for the high shock, there were positive correlations with the standard health questionnaire and with the state trait anxiety. So the higher you scored on the state trait anxiety, the higher the scores on your high intensity painful shock. Now, the most interesting effect actually was when we calculated the effect. So this was just all the high shocks. But when we looked at the induced effect of the placebo or the nocebo suggestions, we only saw a significant correlation with the nocebo suggestion. And that was with somatosensory amplification and with the scale for hypochondria, the Whitley index. In a way that people that showed high somatosensory amplification, and in the previous experiments, we already found that they are less capable of suppressing pain, but they were very able to increase their pain based on the nocebo suggestion. So apparently the ability to activate your CCK system and experience more pain is a tendency we see with people that also are scared of pain, have hypochondriac tendencies, etc. And we have always, so why is this interesting? A little bit of a take-home message or a useful finding. I think in medical science, we have always been fascinated by the placebo effect and potential applications of the placebo effect. But maybe it's far more important to try to avoid a nocebo effect, yeah, or diminish nocebo suggestions, because they might have far more relevant effects on the well-being of the patient than a potential placebo effect. And this actually also matches with observation that if people have been sick for a long time and they get tested, if they get very negative results, so they discover that you actually have a serious disease, they might even become very suddenly even sicker, yeah, than would normally be expected related to this knowledge and nocebo suggestions. Is it then an imagined effect? No, it's a very real physiological effect. So that's the other take-home message. Placebo effects and nocebo effects and conditioned pain modulation are very measurable and very much embedded within our physiological systems. Our immune system responds to it, our heart rate, our breathing, etc. So they do have very real implications for our health and well-being. So again, no correlations with the placebo effect, but a clear correlation between the nocebo effect and our tendencies for pain catastrophizing and anxiety. Yeah. There's one more experiment if you want. This is also what I did a podcast on, the Milgram experiment. Okay, continue with the final experiment. And I think that's also, this is really experimental psychology, where we only use healthy volunteers and we really look only at noxious or nociceptive pain. Yeah, so clearly based on the response. Now, the majority of my position is actually dedicated to education. So I have a very limited research position, and most of the research I do with students for thesis projects, but I do get occasionally invited for special festivals or events to do a demonstration for the public as well. And this was actually my favorite one because we also got it published afterwards. But we were invited in Amsterdam in the Science Museum Nemo. They had a huge science festival that lasted all night to do on the spot a pain experiment. We started at 9 p.m., so 9 in the evening, and collected data till 4 in the morning in 60 participants in 4 conditions, or in the end that was what we had. And to make it interesting for the public, we actually based our experiment on the very famous or infamous Milgram experiments. And I don't know if you're familiar with the Milgram experiments. Maybe it needs a little bit of explanation. Milgram see nobody, I don't see, okay. In the last centuries, I think in the 70s, I'm now looking 70s, 60s, early 70s. They performed a series of experiments in which they invited the participant and told the participant that they were doing a research on memory and learning. And it was their task to act as the teacher, and there was another participant, but I was actually in on the experiment. It was a Confederate, so it was one of the experimenters. That was the student that was sitting in another room. And they say we have the hypothesis that if people are punished if they make a mistake, they will learn better. And as a teacher, it's your task to punish the other participant. So the other participant was sitting in another room and connected also to electrodes. They had to learn words, and if they made a mistake, they would get an electric shock. And every time they made a mistake, they increased the panel, increasing the shocks. Yeah? Now, this participant was actually not a real participant, as said, but one of the experiments, and electrodes were not really attached, but he would just be there on display, and with every increase in shock intensity, scream harder and become more panicky. Even at a level, and then the display would at a certain moment even display a red field saying dangerous, life-threatening. And this fake participant would really start to scream, say, I have a heart condition, I cannot take it anymore, you know? And the other experiment would be looking at the experiment like, look, are you sure this is okay? Yeah, this is all part of the experiment. Just press the button, that's your task. And it was actually an experiment to see how far people would go in in their obedience towards the experimenter in inflicting dangerously high shocks to another person. And it was actually an experiment on obedience, and there's now obviously a lot of criticism, especially ethical considerations with it, because these participants said afterwards they also suffered a lot of, problems about being under a lot of pressure of doing this. But at the time it was really published as about 2/3 of the participants, 63. Oh, you just looked at up. 60s even, yeah. I think about 2/3 of the participants continued with the experiment, delivering potentially fatal shocks, not really, but fake, to the participant. And at that time, it was also part of proof how far people would go, and also in their the power that was there from experimenters in a white lab coat, and being very persuasive in doing that. Now, it's a very, at least if I look around, it's a very well-known experiment. There have been films made about it, documentaries, pictures, and somehow if I tell about experiments, just pain experiments where I give shocks, a lot of people actually come up with this experiment. So I thought, let's turn it around. Let's make it look like a Milgram experiment. Very long introduction. At the start of the experiment, the people really waited, because you will be amazed how fascinated people are to be a participant in a pain experiment. They were actually willing at the festival to stay in line and wait for over an hour to participate in the experiment. And we used that time to also let them in fill out questionnaires. And we had them fill out a questionnaire on psychopathic traits, also because it's quite media genetic, and pain empathy. So psychopathic traits that are also normally distributed, and empathy. Now, the experiment started with, it was a carousel, and there were 4 conditions. So every participant had 4 conditions. The first condition a participant would get is he would see the participant before him or her, and this participant before him, they would both be having EEG. There's one button and one computer with a screen, and then the participant next to the new participant would be connected to a pain electrode, for the electric shock. Now, this participant that was already in the experiment would be asked to press the button, and if the person pressed the button, 1 second later there would be a flashlight on the screen, eliciting also an evoked potential, and again 1 second later there would be the shock. And it was a very brief shock, but pretty high, and before participants participated, they could feel it and decide based on that level if they wanted to participate or not. So this participant had to press 5 times the buttons, giving him or herself 5 times a painful shock. And the real participant was watching this, passively. And after 5 times, the button was moved to this participant. So now it was not only watching, but it was actually delivering the pain 5 times. Now, if you press a button, we can also measure an evoked potential. And this evoked potential will show a certain negative component, the error-related negativity, when you make a mistake, for example. And our idea was, if you press this button in order to hurt somebody else, this will probably also give rise to an error-related negativity because it feels wrong to do so. Yeah? Now, after these passive conditions, watching somebody giving him or herself pain or giving that person pain, they moved and a new participant would come in and they would then give themselves pain or having pain inflicted by somebody else. So only 20 trials, 5 giving yourself pain, 5 getting pain from somebody else, 5 watching somebody giving him or herself pain, 5 inflicting pain on someone else. These were all the students that joined us. They were all co-authors also on the paper. And this is actually what we found. That is, if you look at when you press the button, the response-locked ERP, we did see an event-related negativity, especially over the frontal cortex, what we know also from the literature, and it was increased when you press the button in order to give a shock to somebody else than to yourself. So it feels more wrong to hurt somebody else than to yourself. Then if we look at the pain evoke potential, that is, we have a higher response if we inflict pain to ourselves than if we have pain inflicted by somebody else. So there's a little bit of a discrepancy, right? We don't like to hurt somebody else, it feels wrong, but we feel less pain if somebody else is hurting us than if we do it ourselves. And maybe you recognize that if you have a splinter and you have to get it from your finger, you know, you rather ask somebody else, at least I do. I don't really like it with a needle to try to get it out. Now, when we look at correlations, what we found is that people that scored high on the psychopathic traits did not really show that difference between hurting themselves or somebody else. They did not really care. Yeah. So the difference became really small. And if we look at pain response and we look at people that score high on pain empathy, they showed an increase effect, but only when they hurt themselves. So our pain empathy is most evident when it concerns ourselves. We feel very empathetic for ourselves. Now there's one, because this is the last experiment I wanted to show. But there's one problem with this kind of experimental pain research in the lab. And that is, of course, we work with healthy volunteers, and of course we cannot really randomly take people from the street to join in this kind of experiments. But people that are volunteering to participate in pain experiments are not a very good sample of the general population. There is some kind of self-selection in there. And this is a very interesting study from Leuven and Maastricht, is that there is sampling bias in pain research. And as long as you work with volunteers, I think in general, there are people that also score higher on adventure risk seeking, etc. So and this is something that really should be taken into account. The other possibility of doing pain research is either using non-painful stimuli, like in the pain empathy experiments, or studying patients with chronic pain or pain conditions and see how their pain attitudes are and how their already present pain is being modulated in different situations. However, it's very difficult and challenging to get approval to also study nociceptive pain in pain patients. Yeah, they already have pain, so inflicting additional pain, for example in the placebo-nocebo experiments, poses also ethical questions. But it was just a little bit of an overview over some different kind of experiments that we did over the years, using both self-report and event-related potential, so a neuroimaging method, to study these different aspects of how our brain can modulate our pain experience based on conditioned pain modulation, so the pain inhibits pain, on a placebo or nocebo suggestion, or on levels of pain empathy, and inflicting pain to yourself or somebody else, so the social context. And that was a little bit too early, but there was also a Q&A, I think. So you need to have questions. It's your turn. It's obligatory.
Okay, thank you, Marijtje. Shall we give Marijtje a round of applause for the presentation? Okay, we've got some question in. Maybe we can answer the questions that have been sent to us. So the first question, let's see. Oh, yeah. So one question that got into Mentimeter, the question was related to the ice bucket demo that we did earlier. And Marijtje, the question, Marijtje, not that one. So the question that got into Mentimeter was when we did the ice demo. The question was when we put our hand in, and then we got the response. So to which part of the brain or to which organ the pain is being sent to? So does it, yeah, but which part?
Which part of the brain? The pain matrix is actually quite wide. So that there's a slide on that one. Slides have gone. But in general, the first pain, that also has to do with the localization, goes to the primary somatosensory cortex. The insula is also important. Primary motor cortex get activated. ACC, frontal cortex, basal ganglia, amygdala. They're all involved in receiving this sending information from the body part is being exposed towards the brain.
After that sent to the brain, does it then being sent to other organs?
Yeah, and then you get the reaction of the brain, and then you get the activation of the descending information with activation of the periaqueductal gray and rostroventral medulla and lower brainstem systems. Pretty much the whole brain is involved. And I actually did break up the talk in two parts. So today is the nociceptive pain, the acute pain. Tomorrow in the lecture I will talk more about chronic pain and psychological interventions for chronic pain.
Then we have questions. And then we have questions in the chat room. It's in Bahasa. Would you like to practice your Bahasa, Marijtje? So I think is the questionnaire being given to the sample, the participants, and what age of the participants can be given the questionnaires? Does it matter how we choose our sample in this placebo effect research? And is there a possibility this kind of research can turn the participants into an aggressive individual? That's the question.
I need some more information about the aggressive part of the question because I did not really get that one.
The research turns the research participants, or can it lead the participants into forming an aggressive act after participating in this kind of
I never gave it any thought. I never have experienced it. The other questions are a little bit more straightforward and easy. That's the age. Of course, these kind of experiments we only do again with healthy volunteers above the age of 16. 16 is the new 18. So we do get ethical approval of participants that can sign knowingly their own informed consent. Yeah. These questionnaires are pretty much also for the adult population. If we want to do pain assessment in patient groups that are underaged, so in children and infants, we have adapted fast skills, we have observation skills, etc. So that really depends on the population. For this kind of research with volunteers, it's aimed at adults. And the last part with the aggressive, I think it's also because it's based on volunteer participation. So I've never experienced any aggression, only morbid fascination. People are just really, and it works really well because normally if we are at a festival, and we did about 4 or 5 festivals, people had incredibly high pain tolerance levels on the electric shocks. They went to pretty high levels, higher than during the pilots on the university, because they were often with whole groups of friends, or this is what we think, and they were really competitive, almost like the ice water bucket. I mean, you cannot stay shorter in the ice water bucket than a student. I mean, you're a staff member, so no, just the social context does influence it a lot. Yeah. And no, we checked for that. We checked for that. And it is amazing that there's still actually a small group of fairly sober people showing up. Yeah. People are amazingly honest. We just ask, and we actually also included participants that did take drugs. We also analyzed that. That was on a conditioned pain modulation experiment. What we did find was we had the hypothesis that alcohol diminishes pain sensitivity. And what we found is that this is actually not the case. Alcohol does not lead to higher pain thresholds or higher pain tolerance, but it does increase the activity of the endogenous opioid system. So, as I like to explain, if you are completely drunk and if you fall off your bicycle, it hurts as much as if it would if you were sober. But if you have to get stitches in the hospital afterwards, you don't need to get an analgesia, because with the fall you create a lot of endogenous opioids. We also looked at smokers, and people that use nicotine hardly get any activity in their endogenous opioid system, so they are not very capable of suppressing their pain. That system seems to be broken. And I know from my colleagues at the hospital that many patients with chronic pain smoke. Now, it is not very likely that people are in their late 30s, they get a hernia or lower back pain or whatever, they develop chronic pain and they think, oh, I have chronic pain, I start smoking. That's not very likely. What we actually think is that smokers have a much higher risk in developing chronic pain because this system is being affected and it has clinical relevance. So that was some of the clinical observations.
You have questions? Yeah. I'll give you the mic so everyone online can hear.
So my name is Odit, and I have some question according to the pain management or pain resilience in our body. Because firstly, for the context, I am enthusiast in physical activity, especially in running. And as we know, when we run for a lot of kilometers, we tend to feel pain in our body. Is there, from your research, anything that insight that you can give me to how to manage the pain, especially when since your research the subject was healthy, But when we were running, we were actually hurting our body, and I need to know or get some insight how to manage the pain, especially when the pain was always happening and happening. That's all. Thank you.
Thank you for the question. This is very interesting because it also applies to me.
Yeah. It's actually a really complicated question, because pain in sports is almost like a complete field. Because on the one hand, if we do especially endurance sports like running, we also activate our endogenous opioid system. People can actually get literally addicted to running because of the endorphins. Hurting your body during sports is because we put physiological stress to our system, and it's a warning signal. So, I mean, if you go to the level of your boundaries, you get signals that you're actually damaging tissues as well, and it's a signal to slow down. But what you also see, that if you do sports in a social context, your pain threshold increase. And they've actually done very interesting research in comparing people that do rowing, because that's a sport that you can do both individually and as a team sport. And they have also discovered that your pain thresholds are higher in a team context than alone. So there's a lot of factors that influence it. If you have an important game, what we see is also very often stress analgesia, that you're so focused on winning that you don't feel the pain only after the game. Sometimes you even get quite a severe injury, like a sports injury, that you continue the game with and not notice until after the game. So there are many factors involved, and it's multifactorial, but in general, if you're training and if you are experiencing pain, most of the time it will be a signal to decrease the stress on your system. Yeah. Very addictive. Yeah. Yeah.
Okay. So in other words, that we can have more pain tolerance when we are training or doing everything together more with other person that have similar interest or similar goal to us, right? Yeah.
And I think that's why a lot of people who run, they run in a community together, and probably that one of the helping factor. Do you want to add something, Marijtje, or we can go to the next question?
Well, yeah. Only one thing, and that's keep in mind, because now many questions are focused on how to decrease pain, but keep in mind that, or how can I have more pain, that pain in general in itself is unhealthy. And pain does have an unhealthy effect on your brain. And pain is really something you should take seriously and avoid, but that's more in general.
Thank you. Then we'll go to the questions from people online. From Bachika Laras, media and political social campaign often include portrayals that may induce pain empathy. What do pain research find about the effect of those depictions on human behavior, attitude, or cognition?
Yeah. As said, I mean, these are in general elicit strong emotions. They activate our pain matrix, and any kind of empathy, I think. But this is not my field of expertise. Yes, social psychology, but I can imagine that it would increase feelings of feeling connected and committed and involved in it. But this is really on the field of more social psychology. So I'm a little bit more in doubt. Maybe there's a specialist on that. Social psychology.
There's a social psychologist here, but I hope that quite answer your question, Bachika. The last one is from Ibu Rianti. Women endurance great pain during their lifespan? Is there any contribution towards the female participants and the contribution to the pre and post labor pain? Maybe in further research.
This is a pretty standard question, and the gender differences. And I always have to disappoint, especially women, is in general, men have higher pain thresholds. They have a different physiological make, and if you just compare the numbers in the lab, men have higher pain thresholds. However, women are better at activating in general their endogenous opioid system. So after the initial pain, they are much better in suppressing high levels of pain for a longer period, also during labor.
Okay, thank you, Marijtje, for today. I think our time is up also, and thank you very much, everyone. Oh, sorry, I have to be at the center so everyone can see. Thank you, everyone, for coming offline. And everyone who joins us online, thank you very much. Thank you for sending in your questions. And thank you for hanging there for 2 hours. I hope to see you again in other research talk that our faculty will organize in the future. Thank you very much, Marijtje.